This recorded session is dedicated to our pre-lab for our module on cell division. We have a number of learning outcomes, course and topic outcome that we are hoping to achieve by the end of this session. So, for our course learning outcome number 3 associated to this module, um, we intend to be able to explain how substances move in and out of the cells and throughout the animal body, and basic physiological processes including respiration, cell division, growth, and development. So the emphasis is on the process of cell division. Then for our topic learning outcome number 5, um, at the end of this module, you will be able to understand how different cells grow and divide. So, for this activity, there are two specific objectives um, that you will be observing as you perform the laboratory proper. So, first is to be able to identify the different stages of cell cycle through the observation of our model systems. And you should be able to later on differentiate the stages of mitosis, no? Mitosis and meiosis, but we're going to provide or we're going to give emphasis, or our focus is on mitotic cell division. So let's discuss a few things, a few topics that will reinforce your learning and are required for you to be able to perform the activity. So first, let's take a look at the cell cycle. So the cell cycle, as the name implies, is a process or an event that all cells in an animal body, for the purposes of our course, no, a set of events that cells undergo, and we call it a cycle because um, as the process completes, no, the outputs or the divided cells, the progeny cells, will have to begin the cycle, will have to begin the initial steps of the cycle. No? In animals, it's important as the cell cycle allows growth. No? It allows further development of animals. And simply, we can define cell cycle as the process through which cells replicate and produce progeny or daughter cells. Now, before we look into the key or the visual stages or the stages of mitotic division, which you will be observing in your model systems later on as we do the laboratory proper. No? Let's first take a look at the processes that are, or the stages that are not so visual, that you can really appreciate looking at the microscope or using the microscope. No? First, or the first of these stages is actually the interface, where the cell spends most of its time, and during which it grows. During this time, the cell prepares for replication. The cell prepares for division. It replicates its chromosomes, the DNA is replicated, important material like proteins are produced or synthesized. No? And after interphase, once it's successfully completed and the cell checks that everything is needed, or everything needed are already produced, no, cellularly, then the cell will enter the mitotic cycle or the mitotic division. Okay. Um, there is a series of key events that happen during the cell cycle. So, the initial growth and development of the dividing cell, the synthesis of DNA or replication of DNA to produce two copies that will later on be divided or distributed into the progeny cells. The preparatory steps before division, and the cell then divides. No? So here you can see an illustration of the stages of cell cycle. So as I mentioned, the interphase is the period where the cell stays for longer or for the longest time. So here, the interface is divided into G1, S, and G2. So during G1, cell growth occurs. So the cell will produce all the materials necessary for it to be able to synthesize DNA and be able to undergo mitotic division. During the S phase, DNA is replicated, DNA is copied into two, and then once the DNA in your laboratory proper, you will see um, replicated DNA in form of chromosomes. No? So once the chromosomes are in two sets, they can be distributed to two cells already. The cell will enter the G2 phase where it will prepare for mitosis. Mitosis has four key steps. And helpful for you to remember the acronym PMAT: P prophase, M metaphase, A anaphase, and T telophase. So during the PMAT stages, the chromosomes undergo um, changes in terms of their positioning, in terms of their orientation within the animal cell. And once that telophase is about to complete, the cell will now enter cytokinesis, where it will officially be divided into two. Okay, so we mentioned this already, the interphase and the mitotic phase as the main periods or stages of the cell cycle. Okay, so this one is a good summary diagram connecting the steps or the key stages. So during interphase and mitotic division, or rather cell division proper. No? So aside from mitotic division, which is what we're gonna focus today, no, there's another type of cell division that happens in cells that are gametogenic, or cells that are intended for reproduction, the sperm and the egg, and that is called meiosis. For bodily cells, called somatic cells, mitosis is the appropriate term. So the difference between them is during mitosis, or cells that undergo mitosis will produce daughter cells with the same number of chromosomes as the parent cell. On the other hand, meiosis, the gametogenic cells, the daughter cells do not have the same number of chromosomes as their parent cell. So usually it's halved, because in meiosis, the final product, the daughter cells, the sperm and the egg, should be haploid in nature because they're intended for fertilization, they're intended for reproduction. So the egg should be haploid, the sperm should be haploid. So when they combine, During fertilization, they will produce a zygote that is diploid. So they will somehow reconstitute one another. The egg and the sperm will reconstitute the number of chromosomes for one another. Okay. Also, just to introduce the concept of or the term karyokinesis. So, karyo, nucleus, essentially the division of the nucleus, division of the DNA of the chromosomes. No? Cytokinesis is the visual division or the more visually appreciable step, because you can see that the two cells are really separating, no? Okay, so I mentioned this already. Okay, so here you can see an artist illustration of a cell undergoing interphase. So it's getting bigger, it's preparing the materials needed for DNA replication and for cell division later on. No? So during this time, you can expect accumulation of proteins, accumulation of other biomolecules that are needed for the daughter cells, no? Because think of it this way: the cell will produce two new cells, so there has to be equal components of biomolecules for the two progeny cells. So during the interphase, that synthesis or preparation of biomaterials or biomolecules is happening in our model systems. um, Usually the interface cell looks like this. There's an intact nucleus. The cell is bigger. No, you can appreciate the nucleus, within it, very prominent nuclear membrane, very prominent nucleolar region, and the DNA within the nucleus is highly staining, no, for our for our model systems, for our model cells that you will observe later on in the laboratory proper. Okay, so I mentioned this already, the G1S and G2. Important to note, I think, will be under G2. Two things that we should remember. So before the cell enters the cell division stage, the karyokinesis and cytokinesis stage, we expect at G2 the organelles are already doubled in number and additional cytoplasm is produced. Now, let's take a look at the DNA material, the genetic material, before we proceed to discussing the cell division proper. No, so the DNA, which is a double helix nucleic acid based material. So, as a review, so the DNA of cells are double helix in structure, primarily composed of nucleotide bases, A, G, C, and T. G partners with C, and A partners with T. No? Chemically, you can appreciate the structure here. Right? So there is a backbone, sugar backbone, sugar phosphate backbone. And within this backbone, or in the middle, no, think of it as a ladder. Think of the DNA helix as a ladder. In the middle, the bases, the nucleic acid bases, are connected by hydrogen bonds. Okay. Now, so as I mentioned, during the S phase, no, we expect replication to synthesis of DNA to take place. And each cell after the S phase must be able to produce two sets of DNA, to double the material of the original, double the amount of the original DNA found in the parent cell. Okay, so how does DNA replicate? So it replicates by um, what do you call this, a uh, uh, by the so how it replicates the original strands, the parent strands that are replicated, that are represented in yellow in this illustration, they unwind. Okay, the hydrogen bonds between the original nucleic acid bases, they are dissolved, and the old strand, or the parent strand represented in yellow, serves as a template for the synthesis of a new DNA strand. So think of it this way: the daughter cells, okay, will receive uh, half of their genetic material originally from the parent, and the other half as newly synthesized. Okay? Now, let's take a look at the M phase, but I think we should pause for a while to make sure the recording is not too long.